Hi everyone, my name is Eloise. I'm a postdoc in Dr. Martin and Agipo lab, and today I'm very honored to present our work about whole genome deconvolution unveils Alzheimer resilient epigenetic signature. So, a self-transposed accessible chromatin or ATAC-seq is an effective technique to measure the open chromatin accessibility. The open chromatin regions are nucleosome-depleted regions accessible for gene expression and regulatory processes. ATAC-seq enabled to characterize chromatin state and changes in diseases and investigated the role of coding and non-coding region in biological processes. Historically, ATAC-seq was performed on bulk on a piece of tissue to measure the chromatin accessibility across a population of cells. It is a low cost and simple to perform methods to get a robust measure of the chromatin landscape in disease, and it can be performed on large cohorts. On the other side, recent technique is single-cell ATAC-seq. Single-cell ATAC-seq is an amazing method that can depict the chromatin landscape at a single-cell level. However, it is more expensive, more complex to perform, and also more prone to technical issues such as dropout, making the data analysis more difficult. So here we hypothesize that we could use a machine learning algorithm to reconcile these two techniques and computationally deconvolute and enhance the bulk resolution at the cell type level, providing a robust and low-cost method to detect the cell type-specific open chromatin regions. So to do so, we take inspiration from the source separation problem. The source separation problem is a widely studied problem, especially in computer science and audio processing. And the goal of this problem is to restore or separate individual sources in a mixed signal. As is illustrated here, one classical application can be found in audio processing, and it is a case when multiple instruments are playing all together and only one microphone is recording them. And the goal here is to separate the sound made by drums, guitars, and piano individually. So using the same paradigm here, we would like to separate the chromatin accessibility from different cell type or measure using only one signal, a bulk signal. However, bulk deconvolution is not a new problem at all. It has been widely investigated for the past few years, especially for bulk RNA sequencing. But previous methods focused on predicting the cellular abundance from the bulk sequencing. Following the previous metaphor, this method focused on counting how many pianos, guitars, or drums are playing, but not what are they saying, and prevent from analyzing the differences between the sound made by these instruments. So here we use a machine learning approach to try to solve this problem. However, using machine learning on DNA-based data, such as ATAC-seq, has inherent challenges. First, ATAC-seq, as any DNA-based data, has an extremely large number of sequential features with both short-term and long-term dependencies. Another problem is the scarcity of the data in the context of machine learning, as you may all know. So to solve the first problem, we use the attention mechanism strategy. Attention mechanism is at the basis of any new models, in GPT or any NLP model, and is an extremely powerful method to model long sequences. Briefly, these methods try to extract from the input sequence the information, the most relevant information that lead to the best performances. And in the context of epigenetic, it is highly relevant because in epigenetic, we have like both short-term within the same chromosome interaction, but as well as long-term dependencies due to the 3D geometry of the DNA. To best bet the scarcity of the data, we use another strategy. We generate a synthetic data set from a single nucleus ATAC-seq to create bulk mixture that have a non-composition, but also a realistic ATAC-seq profile. So to do so, we use a single nucleus ATAC-seq from 13 control and use a sampling and aggregating strategy to create cell type specific pseudobulk sample from with a realistic profile by sampling within the same cell type. And then by aggregating this, we create bulk make sure that will be the input of our model. In this way, we can train our model in a supervised fashion to separate the cell type specific profile from the bulk mixture. The advantage of using that is that we can create a combinatorial number of samples to create a data set large enough to be able to train the model. So we derive Self-Former, a model that can deconvolute bulk actually into 6 membrane cell type across the whole genome, predicting more than 41,000 cell type-specific open chromatin regions. We validate our method using a cross-validation strategy and 3 metrics. One metric that measure how realistic is the profile compared to the ground truth, and 2 metrics that measure how well the model can accurately predict an open chromatin region or not. And on these 3 metrics, what we can observe is that our model significantly outperform a previous machine learning baseline used for pressure deconvolution. We also validated our model using an external single nucleus ataxic data set and compared the deconvoluted profile for different cell type with the profile
in addition to high decorrelation
measure using single nucleus. And we observe a significantly high correlation within the same cell type between the 2 modalities. Additionally, we train an external classifier trained to predict the cell type level on single nucleus and apply a deconvoluted signal. And we observe
suggesting that the model
perfect classification, meaning that the model preserves the cell type specific signature. We then apply it on to unveil Alzheimer resilient epigenetic signature. So Alzheimer resilient are people that are cognitively healthy but do have a high pathological load of the disease. And these people are really exciting because they're enabled to because understanding the mechanism that confer protection against cognitive decline in this individual represents a unique therapeutic opportunity. So briefly, we applied this model on a large cohort from different brain regions, and we found that most of the epigenetic signature are localized in the hippocampus, involving memory and learning processes, and imply synaptic processes and inflammatory response. With that, I would like to thank my 2 amazing supervisors, my colleagues, the collaborator, and all the organizers. Thank you very much. And I'm happy to take any questions.
We have time for 1, maybe 2 quick questions. In the back? No? Oh. Wait a second for the online audience. We need the mic. Yeah, I can do this once a day.
Great talk. How does this perform for cellular subtypes? You've got the main cell types there.
Yeah.
Have you investigated subtypes?
Yeah. We try on a more resolved on cellular cell type. The only limitation is the computational power here because we use a deep learning model. So we can go further. We show that it can work on subtype like inhibitory cell type, but at the cost of reducing of other cell types. So we have to choose a panel of 8 or 10 maximum due to computational power again.